All right, guys. So let's continue on this one. So we have two more to talk about. One is protein, and the other one is the nucleic acids. Same thing. They are polymers. So what you need to know is that what are the monomers? Yeah. And for proteins, the monomers are amino acids. So this is the structure of the protein. So this is a linear structure. It's also called primary structure. You can see that it's a string of amino acids. So it's very easy to recognize or to appreciate that this is a polymer from the combination of the monomer. Yeah. So when you have an amino acids, your body put them together. When you eat protein, your body take them apart, you know, uh, uh, hydrolysis, put them apart, then you can absorb the amino acids one at a time into your body. After you take those amino acids, they become the building blocks. Our body put them together to form protein. So when they put them together, it's the very beginning is a linear structure or primary structure. We call these proteins, we have multiple terms to call a protein. When it's large one, and we don't just use this one. We usually have this large one, we fold it, just like a string, and not much you can do about it. So you would knit them together to form a glove, basically. And so that knitting is a structure. We call it folding of this structure. And with that one, we call it protein. After the folding, you have this functional piece. That's a protein. Sometimes we have these with a shorter one, we call it peptide. So peptide could be short. Sometimes when it's short, its structure become not as obvious as you can see because it's very short. Uh, 7 amino acids could be a functional peptide. Yeah. So it's very common. It doesn't have to be very long. A longer one, we call it polypeptide. So basically, short piece put together, we call it polypeptide. And the large one, we call it protein. So that's multiple different name, but they are basically the same thing. The polymers are peptide, polypeptide, protein. The monomers are amino acids. When they put together, it's a linear structure. It's also called primary structure. Our body need to fold it, knitting it, knit it to form a functional piece. And so we will fold it uh, to change from the primary structure to a secondary structure, and then to a tertiary structure to become more look like a piece, to a quaternary structure. Depends on the protein. Sometimes we don't need to fold it. Sometimes we just need a secondary structure. Sometimes we need tertiary structure. Sometimes we need quaternary structure. But what are those structures? Primary structure is easy. It's a linear structure. Secondary structure is two-dimensional in a way, or a simpler form. It basically two form. One is a helix. So it's like, can you see that? Yes. So it's like that. Yeah, I don't know if I have a picture like this. So this is helix. Okay, so it was not string, it's become a coil. Okay, let's do put it that way, a coil of a helix. Or a beta pleated sheath. So basically you can have a, hold on a second. You can have the string by lining, and it will become a piece of the paper sheath. Yeah, so that is a secondary structure. With that second structure, you can further fold it. Like if you have a piece of paper, and it's fine if I can have a paper, like this paper right here, a piece of paper, right? And you can fold it further to become, can you see that? I don't know, blur a little bit, huh? Yeah, to become like like a tube, all right? Like a tube. So this is tertiary structure. Yeah, it's very common. We have this tube, and in a membrane like this one here, we can insert it on the membrane right here. We can insert this on the membrane, so it become a channel. So that's a protein on the membrane. Is a tube, is a tertiary structure. Put it in here. It allow water to go through. It allow ion to go through because ions are water soluble. It cannot go through without a channel. So that channel will be a protein. So that's that tertiary structure. Then how about quaternary structure? Quaternary structure is the way to describe the proteins. It's formed by combination of multiple, more than two protein together. So for example, this is a cup, right? Okay, you can see that you have a cap put together. Yeah, so that's a quaternary structure. You have multiple pieces. So this is example of showing you how does primary structure become a secondary structure. So it's a folding. Okay, you have this linear structure, amino acids 1 to 8, 1 to 9, 9 amino acids. Linear structure, primary structure is also a linear chain, right? It's a chain. You got one chain, fold it, twist it, become a coil. We call it alpha helix. So that's secondary structure. You can have a secondary structure to become tertiary structure, become a ball. So that's tertiary structure. You can have a tertiary structure to become quaternary structure. To make a quaternary structure, it's not just one chain. You need multiple chains, more than one. So you have 1, 2, 3, 4. You have 4 of these chains, 4 of these chains put together to become the finalized protein. So this is a quaternary structure. An example would be hemoglobin. Hemoglobin is a quaternary structure. It has 4 chains. Similar thing, a chain contains heme. Heme unit is one to bind oxygen. So hemoglobin is a very effective oxygen transporter. It can bind on protein, transport proteins, binds on oxygen, transport oxygen throughout our body. So hemoglobin is an oxygen binding protein. So that's that structure. So unique part about the protein is that it's the same thing like other polymer. It has monomer bind to become polymer. However, the unique part about protein is its folding. Yeah, so folding make it unique shape to do multiple things. You fold it to become a globe. So this is a globe, a globular, a globe, which is like you knitting to form a like a wallet. Can people do that? I don't know. But you can knit it to become like a glove, right? Beanie. Yeah, so a beanie, you can put things inside of it. So that's the beauty of the protein. You can fold it in a way that you can pocket it with some other stuff inside of it. So yeah, so that's that. All right. And so the folding is the functional structure. However, when we need to digest it, when you eat the protein, it's folded, uh, you will need to unfold it so you can cut it, digest it, and then you can absorb it. So this unfolded process is called denaturation. So a quick question can be, which of following describe denaturation? It's a protein unfolding process. Yeah, we commonly see this unfolding process in our digestive system. For example, cooking can denature your protein. Heat can denature your protein. pH, change pH, like our acidic, like in your stomach, the acidic environment can cause denaturation. So unfold it so we can digest it. Yeah, cooking and pH change. Yeah, so that's denaturation. Just remember that terminology. So that's the protein. Now, the last one that we are going to talk about is the nucleic acid. Sorry, nucleic acid. So nucleic acids, we have two major nucleic acids that we will talk about. One is called DNA, excuse me, and the other one is called RNA. So these are all genetic related molecule. These molecule store our genetic codes. I personally feel it's very fascinating. Like you, every organism, every cell has a DNA. Every virus has this genetic code. Any like life or beyond life, you have this genetic code. And so it's just like in the era of like digital life, right? Everything is can be digitalized, like your computer, your picture, your document can be digitalized, and each individual can also has some form of this digitalization. It has this code, genetic code. Yeah. So they are stored as a form of DNA and RNA. You have human use DNA and RNA. Most of the life organism have those. Yeah. So we can eat, we can absorb these from taking it from our food, like vegetable or animal, because they can provide proteins, same amino acids, carbohydrates, same carbohydrates, nucleic acids, same nucleic acids. All right, so that's that, nucleic acids. Polymers are DNA, deoxyribonucleic acid, RNA, ribonucleic acid. Yeah, so those are the genetic coding material. So those are polymer. Then what is the monomer? This is the monomer. So this is a monomer. This is called nucleotide. Generic, so it's common like structure, just like generic amino acids. Uh, there are multiple type of amino acids, but and also there are multiple type of this monomer. Put them together, just like multiple type of the sugar, glucose, galactose, fructose, right? Those are multiple form of the monomer, monosaccharides. Same thing here. You have multiple form of this monomer nucleotide. And so that's the structure, like this one here. Two of them put together, multiple of them together become the polymer. So nucleotide is formed by a phosphate group. Sugar is a 5-carbon sugar. So, and then you have the phosphate group, nitrogenous group, nitrogenous base. So that's that. Put together. So this is one of these nucleotide, 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 a string. That's one string, two string, like that. So you have two string with comparable pairing, complementary pairing. These two string will form a structure, and this is what we have with the DNA. So DNA is a polymer of the nucleotides. Is a double string. Is a double string DNA. So two string. Yeah, and with a complementary pairing of these nucleotides. So that's DNA, polymer. This is the RNA. So RNA, again, you have the nucleotides, nucleotides, nucleotides. You don't have two string, you only have single string. So that's the difference between DNA and RNA. So that's that. So here is a summary about the difference between DNA and RNA. DNA is called deoxyribonucleic acid. It's a polymer. RNA is a ribonucleic acid. It's a polymer. The nitrogenous base are different. You have cysteine, guanine, adenine, and uracil. In the in the DNA, you have, excuse me, cysteine, guanine, adenine, tyrosine. Yeah. And the DNA is double strand, RNA is single strand. So that's that. All right. Uh, Let me know if you have any question. Okay. Excuse me. See you next time. Yes.